All right, ladies and gentlemen, let's talk today about DNA replication. Remember, we've looked at the idea of what DNA was, that it is a molecule that is designed to carry information, primarily to make proteins. Well, remember, when we make new cells, because we always started off as a single-celled critter, that zygote, that when we make new cells, we have to copy that DNA, and that's called replication. And if you remember, that occurs during interphase. Remember the S phase, the synthesis phase? This is where this is occurring, in the S phase of interphase. What are we doing in DNA replication? Well, simply put, we're going to start by separating, okay? And I want to show you this real quick. We're going to separate the DNA. See right here where the DNA has been split apart? We're going to form something called forks, replication forks, okay? What we're going to do is we're going to, first off, we're going to separate this two strands of DNA. Because remember, it's two strands. Remember, they go opposite of each other. One goes 5 to 3 this direction, the other one goes 5 to 3 this direction. So we have to first unzip it like a zipper, okay? And we're going to call those things replication forks, okay? And we're going to build new strands of DNA on both sides. Now, how do we do that? Well, first things first, we got to separate these two strands. And when we separate the two strands, we're going through something and going to have to use something called a helicase, an enzyme. It's the thing that's going to unzip the DNA, okay? It's going to do what we saw right here, where the DNA is actually split in the fork. Right in this area right here where it's splitting, there's going to be an enzyme called a helicase, which is going to be unzipping the DNA, okay? So that enzyme unwinds and separates the double helix, the twisted ladder shape, okay? Now, what else do we do? Well, first thing is we also have to have another molecule called a DNA polymerase, okay? DNA polymerase, a polymerase builds a polymer, which is a long chain. And DNA, remember, is a big, long set of chain, right? We have to have this molecule, this enzyme here, that's going to build the new DNA strand on the old DNA strands, okay? And it always builds it in the 5 to 3 direction, okay? So what we have here, here's the DNA polymerase, and what it does is it adds new nucleotides to every spot where it's supposed to be added, okay? So it starts here, and there's something called an RNA primer. We're not overly worried about that, okay? But it puts the DNA, I'm going to blow this up a little bit so you can see it. Here's the nucleotide, the sugar, the phosphate, and the nucleic acid, or the nucleotide here, where the nitrogen base on the nucleotide. Sticks it there, sticks it there, sticks it there, comes in, adds another one, then it'll be another one behind it, then another one, then another one. And what we've done is we have built a new strand. This green strand will be a new strand of DNA. And it's going to be complementary, or the opposite of this original old strand, okay? Now, the thing is, though, you got to remember, this is the nucleotide, right? Phosphate and the sugar deoxyribose, which makes up the backbone, and then one of those 4, cytosine, thymine, adenine, or guanine, one of those 4 nitrogen bases. That's your nucleotide. And remember this as well, that when they join, there's your sugar phosphate backbone, right? And over here's your other sugar phosphate backbone, okay? And what's going to happen is we're going to fill them, remember this, with these nitrogen bases. And always remember, T goes with A, thymine with adenine. Remember Chargaff's rule? T always the opposite side of the A. It's held together by two hydrogen bonds. Guanine with cytosine always, it's going to have 3 hydrogen bonds holding. So again, this is the basic setup, right? Keep that in your brain when we're talking about replication, okay? Now, what we do is we have something called a leading strand, okay? The leading strand is the one that you do first. Well, not first, but it's the one that you're going to do in one strand. All right, you're going to start, and I'm going to show you what I'm talking about here. This black one is the original strand. You're going to build the DNA new strand in the 5 to 3 direction, which will be opposite this way. You're going to be going this way. Well, you start right here, and then you build it, build it, build it, build it, build it, all the way down. And you go straight down. Okay? You're going to build it in one continuous strand. Okay? You start at one end and you keep going all the way down, okay? Now, that's the leading strand, okay? It's the one that you build in one strand, one time, in that 5 to 3 direction. Now, the opposite of that is something called the lagging strand, okay? Here's the lagging strand. This is the other side. We're going to build the lagging strand here as well on the opposite side. Again, it's complementary to the opposite of what this one is. But we do not, because of the way we build it in a 5 to 3 direction, this one can't be built in one strand. We build it in little segments. So right here, the DNA polymerase is building a segment. Right here, we built a segment. And then right back here, we'll keep going. We'll build another segment, and then another segment, and then we'll combine them all together, okay? And again, we're not going to get too heavy in it. You're going to see it with one of the videos that I'm going to post in a little bit more three-dimensional here. You're going to see, or I'm going to leave you a link anyway, more than likely. You'll see what actually does occur, okay? And I don't want to get too awfully heavy in that because I don't want to confuse you too much. But it has to do with the way that you're building them in the 5 to 3 direction. You can't build the lagging strand, start at one end because you're only getting a piece of it at a time, okay? But that's okay. It's not that big of a deal, okay? Now, I have a question for you. It's a real simple question. If I've got this strand of DNA, okay, G, C, G, T, A, T, G, if I want to build the complementary of the opposite strand, okay, the opposite strand of this, what is that opposite strand? What G, C, G, T, A, T, G, what's the opposite DNA strand going to look like, okay? Well, it's actually pretty simple because remember, who bonds with G? That would be C. Who bonds with T? That would be A. So what you do is, when you have G, C, G, T, A, T, G, you're going to build the opposite, the complementary strand is going to look like this. C, G, C, A, T, A, C. And you can see these are complementary because guanine always connects with cytosine and thymine always connects with adenine. So you've literally built the reverse side of that, okay? That's the complementary strand. So when DNA has an original strand like this up here, oops, there we go. Right? Here's my original strand right here. I'm going to build the complementary strand this way, okay? I'm going to put everything the exact opposite of what it should be from the original. And then by the time we get done, we got a complete DNA molecule right here, okay? So, I simply put, and you'll have these notes on the setup that I'm going to send you a little bit, and you'll have a video link to another one, and then you'll see this video on my YouTube channel. This is the basics of DNA replication, keeping it simple, right? Now, if you have any questions you need between now and the next time we meet, you need to make sure you send me an email, okay? Don't hesitate to send me an email asking me a question, okay? Please don't forget that, because if you do, you're going to get behind, and I don't want you to do that. So if you have any questions, don't hesitate to ask, okay? All right, so that's DNA replication, right? That's the copying of DNA.